Electrical pacemaker activity of cardiac autorhythmic cells by Hyla Torres. As far back as ancient texts go, humans have known the importance of the heart to the human body. Scholars of ancient Egyptian, Greek, Roman, and even Chinese history have written about beliefs of what the heart does for the body. So go back to sixth grade, think about those ancient civilizations. They were right in noting that it works as a pump for the body. Later on, Aristotle initially wrote of his beliefs of the heart associated with emotion, and many people during the Middle Ages believed what he had said. So this was sort of a step back, so to speak. Knowledge of the human cardiovascular anatomy over the next few centuries thankfully increased as we learned more and developed new skills in scientific discovery of what we know today as cardiology. The cardiovascular system is made up of the heart and all the blood vessels of the human body. The heart is an organ made up of tissue, muscular tissue, that functions to constantly pump blood flowing through the body throughout a person's entire lifetime. The heart has the ability to work with blood vessels for delivery of this oxygen rich blood to every body organ and back again to the heart to do it all over again. What is unique about the heart is that it is constantly working, as I just mentioned. Sometimes it is working hard and fast, and other times much slower. But what remains the same is the need to keep the person alive with its functions. So, today, class, we will be diving into an area of the circulatory system that are the cells that make up the heart and its components of the electrical pacemaker activities within the cardiac autorhythmic cells. To dig a little deeper into our topic today, we are going to specifically be looking at the electrical pacemaker activity of cardiac autorhythmic cells. The heart beats and contracts without any nervous input. How is this possible? Well, specialized pacemaker cells. To ensure we're all on the same page, let's recall some of these definitions. Let's start by recalling that a pacemaker is a system that has the ability to send electrical signals or impulses directly to the heart to keep its beat, parts of the heart. There are biological pacemakers of the heart that are naturally made in us, and there are people who have needs for an electric pacemaker when their system has dysfunctioned or failed them in some sort of way. But that's a topic for another day. Recall that the natural pacemakers are in the cardiac system found in the sinus node of the heart. This is where the heartbeats are derived. The system here has the ability to produce the electrical impulses in the heart muscle, which in turn will pressure the heart to pump blood. For those that need an electrical pacemaker, they're designed to always be pumping to stimulate the heart at a fixed rate, or even at the ability, they even have the ability to elevate the rate. So, during exercise, for example. Some pacemakers have been designed to even detect heartbeats that have too long of a pause in between beats. Moving on to define the cardioautorhythmic cells, these are commonly referred to as the pacemakers of the heart. These cells have the ability to synchronize cells for contractions of the heart. Think about the root word that you see there, auto. It just automatically does it. And then rhythmic, rhythm. So think about that. These are the pacemakers that we are biologically designed with. This is actually what makes these cells unique compared to, let's say, skeletal or even smooth muscle cells. Cardiac muscle cells only have one nucleus, they're striated, the component of intercalated discs, and they are branched. What does this mean? So I'm just noticing or stating some things that you can notice about it. The branched attributes gives it its strength. These are very important to recognize, and It's important. It's crucial for the heart to be strong because think about it, it's always pumping. We take this for granted. The human body needs to be resilient as possible to things like stress or exercise and so on. We need our hearts to be strong and to have the ability to pick up the pace when we need it to. Think about, everything that we learned previously with the nervous system and what happens when our bodies go into fight or flight. What would happen if we didn't have those changes happening in our body, happening with our heart rate? Just something to think about. Um, with skeletal muscles, tears can happen when you're injured. Think of a sports related injury. Heart muscle tears, not as common, and that is for a good reason. Digging in more to the cells of the heart, there are two types of cardiac muscle cells. There's autorhythmic cells and contractile cells. A large majority of cardiac muscle cells are found to be contractile. Some say it's about 99%. Action potentials for contractile cells come from gap junctions of intercalated discs, and this comes from the autorhythmic cells. We're going to keep talking about that. You can see on the image here with just a glance the difference in how the graphs look between these two types of cells. We've got autorhythmic cells on the left, contractile cells on the right. So you can see the difference between the two. We're gonna go over why this is, what this means, what we're looking for here in just a moment. Talking more specifically to the autorhythmic cells, they do not have a resting potential. They have channels called IF channels, and the F stands for funny. You've probably heard this term before when it comes to the cardiovascular system. Funny just means that they are leaky. These channels leak things like sodium and potassium. This is when the pacemaker potential occurs. Once enough sodium and potassium enters the cell through these channels, threshold is reached. Soon the calcium channels will begin to open, and we know that calcium encourages a positive charge. Next, repolarization will begin and potassium channels open. They are what generate the action potentials, even though autorhythmic cells only make up a small percentage of cardiac cells. When I say small, I mean about 1%. So as we said, contractile cells, about 99%, autorhythmic cells, only about 1%, so a very small amount. Looking at the electrical conduction system, we see how the autorhythmic cells are arranged throughout the heart. How will such a small amount of cells be dispersed across the heart? Think about that for a minute. I like to think of it like an important game. I'm not really a sports person, but we all are pretty familiar with the Super Bowl, for example. When it's game day for the Super Bowl, I'm not going to just let every player go out on the field all willy-nilly wherever they want. They're going to be placed there strategically. We're going to be putting people in their places, their strong points, where they're going to do their best functioning. So the same thing goes with the heart here. The cells in the heart need to be in specific places. So let's dig into where they are exactly. We're going to begin by looking at the SA node, the sinoatrial node. It's displayed right here in red. You can see that red in the middle, SAN. Again, remember, these are the pacemakers of the heart. These cells have pacemaker potential. They play a role in heart contraction conduction. The SA node controls the beats per minute and is responsible for giving the direction to other anatomical parts of the cardiac anatomy. So that's where we're beginning. In order to maintain efficiency, the SA node gets assistance on this from the internodal pathways and the AV nodes, or it's showed here in blue, the AVN. The SA node itself is simply a specialized group of cells that themselves do not possess the ability to contract, but they do automatically send impulses to the other parts of the heart to tell them to contract. The internodal pathways have the ability to go to all of the atrial muscles of the heart. So to clarify, the SA node works when the action potential is fired and it spreads to the other atria. When an action potential is fired, you will notice the contractile cells that follow directions on how many beats per minute they should be producing. The other place you can find these cells in the bundle of His and the Purkinje fibers, also noted in this diagram towards the bottom. These fibers are spread. They spread from the AV node out and around the heart. They work to spread the signals given to other parts of the ventricles. You can see on the diagram of the heart where it's red, that's the area that it's spreading. It's really hard to see two-dimensionally. It's definitely more of a three-dimensional concept, but you get the idea looking at the image here. So yes, the bundle of His and the Purkinje fibers, they work together as a team for the greater good of the body's electrical conduction system. You can think of this as a little team here, the SA node, the AV node, and the bundle of His with the Purkinje fibers. And that's how it moves. So let's talk about a real life application here. In a recent study, the effects of N-acetyl-L-cysteine, also known as NAC, was studied on the beats of frog atria. So not in humans, but we're looking at frogs here in vivo. NAC is a compound that is commonly used in fortification of tissues to defend against everyday environmental pollutants. Think about things that you might be exposed to. You got a long commute, maybe you're exposed to some smog. You're around a lot of smokers, maybe you're inhaling things that way. So anyways, NAC in this study was shown to affect how when the frog was in the saline solution, the atria beat impulsively. The figures drawn displayed how the atria cells were actually more respondent to NAC than they were with the autorhythmic cells themselves. Why is that? Let's talk about what NAC is. The N-acetyl-L-cysteine is a molecule that is easily taken up by other cells due to its thiol contents, and it has a low molecular weight. So that's what helps it be so easily taken up by other cells. It has the ability to be hydrolyzed quickly and will release the cysteine that predecessors glutathione, which has properties to protect against damage from oxidative stressors. So here we go. That's how it's happening. That's how it's been protecting against these environmental pollutants. They have the ability, the NAC has the ability to fight off toxicity from heavy metals in the system as well. Additionally, it can be joined with other agents to inverse oxidative stressors. In this particular study in vivo, there were various concentrations of NAC solutions used, so they did small amounts, some middle ground amounts, some large amounts, and with these various solutions, they proved to prevent types of necrosis, the environmental pollutants, they protected against that, and then the various types of heavy metals exposed to. The study concluded by stating NAC has various and strange effects on the in vivo frog atria, and there was a certain amount of concentration functioning that actually inhibited the atria contractions completely. This shows the cells are more sensitive to the NAC than to the autorhythmic cells, which have the power to generate the action potential of the pacemakers, as we just mentioned. Just giving you something to think about. We're not frogs, but you know, systems are similar in that way. They've got those autorhythmic cells. Take a look at the image displayed on the screen here. It's showing the atria tension. It includes the contractions, breath, even the potentials of the transmembrane of the atria. That's in D. You can see in figure D towards the bottom, the atrium, the ventricles here, A, B, and C. That's going to show you the charts there. So just something to think about. It's always good to look at the examples, the real life examples of how this is working. So when next time you're slowing down, you're thinking about your heartbeat, or perhaps you've just finished an exercise and you're checking your pulse, start thinking about that. Think about your autorhythmic cells. Maybe show them some gratitude. Just kidding. But it is something to think about. Thank you.